Good morning, everyone. This is Ma'am Hingaban, your grade 7 teacher at Dasmariñas East Integrated High School. Welcome to our second quarter. On this quarter, we're going to talk about biology. And our first topic in this quarter is microscope. What are the parts and the functions of a microscope? The objectives. Number 1, identify parts of the microscope and its function. Number 2, focus specimen using the compound microscope. What is microscope? The word microscope comes from the Latin microscopium, which is derived from the Greek word micros, meaning small, and scoping, meaning to look at. A microscope is a laboratory instrument, which is used to examine or study to see objects that are too small to be seen by the naked eye. A microscope is an instrument used to magnify small objects. In the laboratory, microscopes are used to visualize minute objects such as plant cell, animal cell, bacteria, fungi, and other objects. What is a compound microscope? A compound microscope is a laboratory instrument with high magnification power, which is used to consist of more than one lenses. A compound microscope are used for the study of structural details of a cell, tissue, or organs in section. A compound microscope can magnify the image of a tiny object up to 1,000 times. The term compound means multiple or complex. The compound microscope consists of two lenses, which includes the objective lenses, typically 4 times, 10 times, 40 times, or 100 times. In a rotating nosepiece, the closer the specimen and the eyepiece lens. Mechanism of a compound microscope. A compound microscope creates an image of a specimen by the following steps. First, a specimen is placed between the objective and the condenser. Number 2, the light emitted from the light source is pointed over to the specimen with the help of a condenser lens. Number 3, after that, the light is passed through the specimen and comes towards to the objective lens. Number 4, the objective lens captures the light coming from the specimen and creates a magnified image of the specimen, which is called the primary image. Number 5, the objective lens passes the image through the body tube to the ocular lens or the eyepiece and again magnifies the image. At last, the viewer can see a clear and magnified image of the specimen through the eyepiece. Number 7, occasionally or during the use of 100 times objective lens, oil immersion method is used to produce a highly magnified image of the specimen. In this method, a drop of immersion oil is placed between the objective lens and specimen slide. Structural parts of the microscope and their functions. The structural parts of the microscope provides supports and connecting all the components of the microscope. These are the present 3 important structural parts of the microscope, such as the head, the arm, and the base. The head. It is located at the upper portion of the microscope. The head portion of the microscope is also known as the body tube. Its application is, it connects the eyepiece to the objective lenses and contains the eyepiece. So what is an eyepiece? An eyepiece is also known as an ocular, which is located at the top of the microscope. It views or it sees the specimen through it. The second one, the arm. It refers to the holding portion of the microscope, which is used to carry the microscope. The function of the arm is, it supports the head or the body tube and connects it to the base of the microscope. And the third one, the base. The bottom portion of the microscope on which the arm portion is standing. It holds all the essential components. Its application is, it provides support to the microscope. Eyepiece tube. The eyepiece tube carries the eyepiece lens. It holds the eyepiece at the right place that aligns the perfect within the objective lenses. It also places the eyepiece and the objective lenses within the distance range, generally in focus images. Binocular microscope has two eyepieces that allows you to see both with your eyes. The eyepiece tube is flexible and can be rotated or adjusted to fit the user's distance between the two eyes. A trinocular microscope has an additional third eyepiece tube for connecting a microscope camera. Objective lenses. Objective lenses are the primary optical lens for specimen visualization on a microscope. Objective lenses collect the light, pass through the specimen, and focus the light beam to form a magnified image. The objective lenses are the most important part of the microscope. Objective lenses. So the objective lenses with a higher magnification are usually longer. As the result, the tip of high magnification objective lenses is very close to the specimen. Please be very careful when viewing and handling high magnification objective lenses. Usually, a compound microscope comes with 3 or 4 objective lenses. The most common setting is the scanning objective lens, which is 4 times. A scanning objective lens provides the lowest magnification power of all objective lenses. A scanning objective lens comes from the fact that they provide observers with enough magnification for a wide overview of the slide, essentially with a scan on the side. Low power objective, or 10 times. The low power objective lens has more magnification power than the scanning objective lens, and it is one of the most helpful lenses for general viewing purposes. The high power objectives, or 40 times. The high power objective lens, also known as the high dry lens, is ideal for observing fine details within a specimen sample. Oil immersion objective lens, or 1000 times. The oil immersion objective lens provides the most powerful magnification. However, the refractive index of air and your glass slides are slightly different. So a special immersion oil must be added to bridge the gap. Without the immersion oil, the 100 times lens will not function correctly. The specimen appears blurry and will not achieve an ideal magnification or resolution. Nosepiece. Nosepiece is also known as the revolving turret. Nosepiece is a circular structure housing the objective lenses. There are holes where the objective lenses are screwed in. The nosepiece can be rotated to easily adjust or change the next objective lenses. Specimen stage, or the stage. The stage is a platform that supports the slide. The stage has an opening called aperture for the illuminating beam of light to pass through. The stage clip holds the slides in place. Coarse and fine knobs. 2 adjustment knobs are used to focus the microscope. Fine adjustment knob and coarse adjustment knob. Both knobs can move and stage up and down. You should use the coarse focus knob to bring the specimen into approximate or near focus. Then you use the fine knob to sharpen the focus quality of the image. Condenser. Condensers are lenses that are used to collect and focus light from the illuminator into the specimen. A condenser can be found under the stage, often in conjunction with an iris diaphragm. Illuminator. Illuminator is the light source of a microscope, typically located in the base of the microscope. Mirrors are sometimes used instead of built-in light. Mirrors are used to reflect light from an external light source up through the bottom of the stage. Please always lift it by holding both the arm and the base simultaneously, like this. Have a blessed day. God bless.